Welcome back, everyone. So we're going to continue on with Chapter 3, Cell Structure and Function. So now that we understand how cells are studied and we know the difference between a prokaryotic cell and a eukaryotic cell, we're going to focus in strictly on eukaryotic cells now. So by the end of this chapter, you should be able to describe the structure of eukaryotic plant and animal cells, as well as state the role of the plasma membrane. You should be able to summarize the functions of the major cell organelles, as well as describe the cytoskeleton and the extracellular matrix. Okay, so this is going to be a little bit longer of a video, so just make sure that you leave some time. Okay. So to start us out, let's just do a brief review of the difference between a prokaryotic cell and a eukaryotic cell. So we already know that the prokaryotic cell is a lot smaller and simpler than a eukaryotic cell. So eukaryotic cells, think large and complex. And we know that the prokaryotic cell lacks a nucleus because pro means before, karyon means nucleus. So it's doesn't have a nucleus, so instead the DNA is found in the nucleoid region of the cell, whereas the eukaryotic cell, U means true, so it has that true membrane-bound nucleus that houses the DNA. In addition to a nucleus, the eukaryotic cell has other membrane-bound organelles that perform different functions within the cell, whereas the prokaryotic cell does not have organelles. So basically it's small and simple, and it performs all of its functions within the cytoplasm of the cell. The eukaryotic cell has different organelles to perform those specialized functions, making it a large, complex cell. And then we already talked about the three domains. The prokaryotic cell containing the domains bacteria and archaea, and then the eukaryotic cell containing the domain eukarya, and then further classifying the domain eukarya into the kingdoms plantae, animalia, and fungi, and then the catch-all group, which contains multiple kingdoms, but we just classify it as the protists. Okay, so that's a good summary of our cell review. All right. So we're going to start out with just looking at two eukaryotic cells. So we have a plant cell and an animal cell. So first off, which one do you think is which? Well, give you a hint. The green one is the plant cell. So the plant cells are green because they photosynthesize, right? So what I'd like you to do is take a look at these two cells and compare the plant cell to the animal cell, and take a look and see which structures the plant cell has that the animal cell does not. So one thing that stands out right away is look at how the plant cell has that extra structure outside of the cell membrane. So animal cells have the plasma membrane that all cells have, but the plant cell has a cell wall that surrounds it. So that's something that the animal cell is lacking. And connecting plant cells to other plant cells is plasmodesmata. So that's something that, again, animal cells don't have. So that's one thing that might stand out. Also, looking at color, do you notice anything green that sticks out? You might notice the chloroplasts. So the chloroplast, something we don't have because we don't photosynthesize. Okay. What else do plants have that animals do not? You might notice this large structure in the center called the central vacuole. We have small vacuoles, but we don't have a large central vacuole. You also might notice that plants, they have little plastids, which we don't have. So plastids are something that's unique for plants because they store pigments. Okay. And I think that's about it. So plants, they have several characteristics that animals, or extra structures that animal cells do not. Now flipping that, what is something that the animal cell has that the plant cell does not? Okay. Now you might think right away, hey, look at this nucleus has all this stuff. Well, the plant cell has the same structures inside the nucleus, it's just not listed. And same with the cytoskeleton. It also has microtubules and intermediate stuff. It just doesn't have it listed. But one thing you'll notice under the cytoskeleton is that it's missing something called a centrosome. Okay, so animals have centrosomes, which is a specialized structure that the plant cell lacks. Okay, what else do you see that's something that is missing from the plant cell? If you see here, we have, instead of these little plastids, we have something else the same color, but it's something different called a lysosome. So lysosome, we'll talk more about that. So that's a structure that plants lack that animals contain. And anything else? Okay, I think that's about it on here. All right. Okay, so we're going to start us out with, we're going to watch an Amoeba Sisters video that will give us a good introduction into the different parts of the eukaryotic cell. So it'll go through some of the major organelles and describe them. And then, of course, we'll go through each one individually.
If you had to think back about the most exciting day you ever had in the science classroom, which day would that be? Now, looking back through the years, we have a few. The time we participated in an earthworm dissection, the time we took apart an owl pellet, the osmosis eggs, all of the fruit flies and genetic experiments. Oh, I could go on. But I will never forget one day in my 9th grade science class. My teacher brought in pond water. And I put one drop of pond water on a microscope slide and saw the most amazing thing ever. I saw an amoeba, a single-celled amoeba on that microscope slide, and I was forever stuck on science from that point on. Because I could not believe that this little cell was there, alive on this slide, still eating, because that's what amoebas do a lot, but to imagine that every person is actually made of billions of cells. Of course, not amoeba cells, but animal cells. Billions of animal cells. That's fascinating. In fact, it really makes you reflect on some of the incredible statements of the modern cell theory. The modern cell theory includes the following. First, that the cell is the smallest living unit in all organisms. And second, that all living things are made of cells, one or more cells. The amoeba I observed was a single-celled organism, so unicellular. Humans are made of many cells, so multicellular. And third, all cells come from other pre-existing cells. You know, cells have their own little world inside them. They carry genetic information, they can divide, many have functions and processes that their organelles, structures inside them, can take care of. On our planet, we can divide cells into two major groups. As a cell, you're either a prokaryote or a eukaryote. Bacteria and archaea are prokaryotes, but everything else, plants, animals, fungi, protists, are eukaryotes. Both prokaryotes and eukaryotes have genetic material. Both have cytoplasm. Both have ribosomes, which are small organelles that make protein. Both have cell membranes, which control what goes in and out of the cell. But what makes them different is a big deal. Prokaryote, the pro rhymes with no, and they have no nucleus, which holds the genetic material and controls the cell's activities. Prokaryotes have no membrane-bound organelles. Membrane-bound organelles are fancy organelles like the nucleus and mitochondria and Golgi apparatus. Eukaryotes, the U rhymes with do, they do have membrane-bound organelles. So now you may be wondering, well, what do the organelles do? What are their functions? Well, you know our style. We love our science with a side of comics. So we want to take you on a tour of the ride of your life into the inside of a cell. Now, to start our trip, we're going to have to get through this cell membrane also called a plasma membrane. It's selectively permeable, which means that it'll only let certain select materials in and out, but by doing so, it keeps things in the cells stable, also known as keeping homeostasis. And we have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video because it's very complex and its organization can vary depending on what kind of cell you're looking at. Moving through this cytoplasm, let's start with ribosomes. They are not membrane-bound organelles and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which we'll talk about a little bit later. We're now going to focus on organelles that will be membrane-bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. And the nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. And there's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Now, molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids. Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might start to wonder, what's powering this thing? The mighty mitochondria, or mitochondrion, if you're just talking about one. They're like power plants. The mitochondria make ATP energy in a process called cellular respiration. Now, it's not the type of power plant that you might think of. It runs on glucose, which is a sugar, and it needs the presence of oxygen to efficiently make ATP energy. Now, at this point, we need to mention that eukaryotes are not a one-size-fits-all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, plant cells not only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to even have a green look to them because they have a pigment that captures the light energy and it reflects green light. Now, both plant and animal cells can have vacuoles. Now, vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells can have one large vacuole called a central vacuole, while animal cells can have smaller, several vacuoles. Now, remember how we already said that all cells have membranes? They do. But plant cells additionally can have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not. Okay, now how do we get out of this animal cell that we've been in? Well, we could get out like a protein would. So if we were a protein, we would only be made because of instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome because ribosomes make protein. The ribosomes could be attached to the rough ER, and the rough ER highway would provide a vesicle to send us to the Golgi apparatus where sorting can take place. And if we're tagged for being secreted, we could be sent off through a vesicle from the Golgi to the membrane. Out we go! Now just keep in mind that in our quick tour, there are still so many more awesome organelles found in lots of different types of eukaryotic cells to continue exploring, so to the Google for more. Well, that's it for the Viva Sisters, and we remind you to stay curious.
Okay, so that was, an excellent way that we can kind of, introduce ourselves to the different organelles of the cell, as well as a good review of, the four components of all cells. When we are talking about cells, the components of the cell, it's a good idea to try to stay organized as we go through, and I highly suggest that you make a chart with your notes, and with the chart have different columns. So with one of the columns, and I apologize, I'm writing this with a mouse, maybe title one of the columns organelles, organelle. So you'll write, the different organelle that we're describing, and then the function of the organelle. So that's one column, the function. Okay, and then maybe a drawing of it. Okay. So this is how I recommend that you organize your notes. Okay. So that way, we'll start out with like the plasma membrane. So you'll have the plasma membrane, you'll have its function, and then you can do a drawing. Now, the reason why I highly suggest that you draw a picture of it is because it'll help commit to memory the structure of it. Because sometimes, remember, structure equals function. So if you recognize, for example, with the plasma membrane, you'll see that it's a phospholipid bilayer with different proteins in between and stuff like that. So it'll help you remember some of the important parts of it which contribute to its function. Okay, so so this is again recommended, but of course, you can do whatever you want with yours. All right. So let's move to the first organelle. Actually, this is an organelle. This is just, the membrane that, the plasma membrane that separates the inside of the cell from the outside. So the plasma membrane, of course, the function is to separate the internal contents of the cell from the external contents of the cell. So when we look at this cell membrane, it looks very complex, and we'll actually devote an entire video on the complexity of this plasma membrane. But for now, we're just going to discuss the basics. So we talked about in our last week in our macromolecules video that our plasma membrane is composed of a phospholipid bilayer. So remember that phospholipid lecture. So the phospholipid is has a hydrophilic head, so that's the little ball part. So that's where that phosphate group is, and then a fatty acid tail. That's the hydrophobic part. And you can see that it's a bilayer, so two layers. So we have one phospholipid, and then here's the other phospholipid. So there's two layers of phospholipids, and you can see how they are facing, the fatty acids are facing each other because they're nonpolar, so they're hydrophobic. So they're facing each other to avoid the water or the liquid part. Okay, So this part is the outside of the cell. Yeah, so this, I had to double check here. So this is the outside of the cell, this is the inside of the cell. And then this is the actual plasma membrane here. So you can see how it's keeping the inside of the cell inside, the outside of the cell outside. But it's semi-permeable, meaning that it can allow some substances to come into the cell, but not everything, okay? So we do have these, what are called transmembrane proteins or integral membrane proteins that can allow some things to pass through that are larger, okay? You can see that we also have other structures embedded, so embedded proteins, there's like little cholesterol. So, and we'll talk more about the specific structure of the plasma membrane in the next video. Okay, so for now, we know that it's a phospholipid bilayer embedded with proteins and other things. Okay, and it's semi-permeable, so it allows certain things to pass through, but not everything. All right, so that's our plasma membrane. Moving on, we have the cytoplasm. So the cytoplasm, remember from having all cells contain the cytoplasm. So the cytoplasm basically is the contents between the plasma membrane and the nuclear envelope. And it's mostly made of water. It's about 70 to 80% water, but it's like a jelly-like substance, so it's kind of semi-solid. And the reason why it's semi-solid is because it's not just water. There's other things in there. So it consists of proteins. It has other things in there, such as carbohydrates like our glucose and polysaccharides. There's nucleic acids in there, there's fatty acids, there's glycerol, there's amino acids, there's ions, there's, you know, little different nutrients in there. So basically stuff that enters the plasma membrane, you know, it enters the cytoplasm, right? So it's floating around in there. So that's why the cytoplasm is semi-solid consistency. It's jelly-like. So it's not just a liquid. All right. So it contains all sorts of stuff suspended within, kind of like how your blood is not just liquid, right? I mean, yeah, there's liquid in it, but there's also other things in there. There's, you know, your red blood cells, your white blood cells, your nutrients, there's waste products, that kind of thing. Okay. All right, so that's the cytoplasm, and that one I don't think you can draw. You can just draw a cell, and then it's the stuff, you know, between the plasma membrane and basically the nucleus. You know, anything that's inside that's not an organelle, that's jelly-like stuff. All right, so moving on to the cytoskeleton. So that word cytoskeleton kind of gives away what the function is. So cyto means cell, skeleton. Well, what does the skeleton do to you? It helps hold you in place, right? So it kind of does a number of things. It helps maintain the shape of you. So the cytoskeleton helps maintain the shape of the cell. It also provides structure for the cell. And it's also involved with movement, okay? So shape, structure, and movement are the main functions of the cytoskeleton, okay? So when we're discussing the cytoskeleton, we can kind of divide it into three types of cytoskeleton. So we have the microfilaments, the intermediate filaments, and the microtubules. And these kind of go from smallest or thinnest to thickest. So the microfilaments are the thinnest, and the microtubules are the thickest, with the intermediate being intermediate, in between. Okay, so let's start out by, first of all, let's look at these drawings because they're kind of confusing. So just imagine like the plasma membrane is just outside, okay, of these. So let's start with the microfilaments. So first of all, where are the microfilaments in this picture? There they are. So imagine again that the plasma membrane is just outside. So they are right near the plasma membrane. And their job, it says here, they're the thinnest, and their job is in helping moving cellular components. So they are, these thin, really thin cytoskeletal fibers, and they're going to be helping with moving cellular components. And then it says, for example, cellular division. So imagine we're going to talk about cellular division in chapters 9 and 10. So every time your cell divides, it needs to also, in addition to dividing like the genetic information, which we'll go into detail in chapters 9 and 10, it also needs to like evenly divide like all the, mitochondria and all the organelles and things. So the microfilaments kind of help that out, okay? So it's helping to, divide all of the little components inside of the cell. So that is part of the function of those microfilaments, to help move cellular components around, especially during cellular division. They also are involved with helping maintain the structure of the plasma, the structure of the cell for the plasma membrane. So when we have something like microvilli, which are these little extensive folding that takes place in some of our cells, particularly like the cells in your small intestine. So imagine like your small intestines, the function of your small intestine is to absorb nutrients, right? And so the cells of your small intestines need really big surface areas to maximize the absorption of the nutrients from the food that you eat. And so to do that, we have to have the plasma membrane have extensive folding. And so the microfilaments allow for that extensive folding. So it's providing that structure for that extensive folding. Okay, so the structure of the microvilli are due to those microfilaments. Okay. And then they're also important components of muscle cells. They help in muscle contraction and things like that. So microfilaments are pretty important components of our cytoskeleton. Okay. Next, we have the intermediate filaments, which again, they're called intermediate because they're intermediate in size. And they provide more of a structural function. So they help maintain the structure of the cell so that the cell's not kind of collapsing on itself. So you imagine again, here's the plasma membrane surrounding. And so it keeps the structure of the cell. And it also helps keep all of the organelles in place. So you don't want like the mitochondria floating into the Golgi apparatus or something like that. So it helps maintain the cell shape as well as anchors the organelles in place. Another important function of the intermediate filaments is it can be used for like strength. So for example, if you touch your hair right now, your hair is made of keratin. So the keratin is a compound that strengthens your hair and nails, and that's a form of a type of intermediate filament. Okay, so keratin is found in your hair, which is, one type of intermediate filament. Okay, so that serves as a structural function. All right. And then the final part of our cytoskeleton is the microtubules. So the microtubules are the thickest, but they're also hollow. Okay, so they're not exactly like, they might be, thick compared to the microfilaments, but they are also hollow inside. So these thick microtubules, they can dissolve and reform quickly. So they're useful in helping guide movement of organelles. So this is particularly important when we're talking about cellular division, which again, we'll get into in chapter 9 and 10. So you can see here, like these are little microtubules that are forming. Imagine the cell is about to divide, and attached to the ends of these microtubules are your chromosomes. So what are chromosomes? We'll talk about this a little bit later on with the nucleus. Chromosomes contain your DNA, so it contains segments of DNA. And for humans, we have a total of 46 chromosomes. So these segments of DNA, when your cell divides, we need to divide, we need to make sure that each daughter cell has the correct number of chromosomes. And so these microtubules are important in attaching to the chromosomes so that they get pulled in the right direction so that they end up in the daughter cell correctly. Okay, we don't want to have a cell end up with too many chromosomes and a cell end up with not enough chromosomes. Okay, so they help with our chromosomes moving where they're supposed to go, so they're very important in that regard. They're also important in movement of the cell itself. So we'll talk about in the next slide, some cells have extra components like flagellum, which is like a little tail, so think of a sperm cell, or cilia, which are these little hair-like projections. Those are used for movement, and they are made of the microtubules. Okay, so they're used for movement. All right. Okay, so that's our cytoskeleton with our three main components. So microfilaments used for kind of maintaining the cellular moving cellular components, as well as maintaining like the structure of the microvilli and things like that. Intermediate filaments, shape and structural components, and then the microtubules, guidance of organelles, as well as movement of the cell itself. And then we have the flagella and the cilia. So the flagella and the cilia, again, are not found in all cells. So this is kind of an optional structure that some of our eukaryotic cells do have. So the flagella, or flagellum singular, is just a tail-like structure that will stick out from the plasma membrane. So here's the cell down here. And it's used for locomotion, so it's used for movement. So think of a sperm cell. So that sperm cell basically just has a large head that contains the nucleus with the package, the DNA that needs to make it to the egg. And so to propel that important DNA package, we need to have that tail for movement. Okay. The cilia, or cilium singular, is these little hair-like projections, and they're also used for movement, so they could be used for movement of the entire organism. So think of like a little paramecium, a single-celled protist. So they have these little hair-like projections that allow them to move. Or it could be part of like a static cell. So think about like the cells that line your trachea, your windpipe. So if you're coughing up something, you have stuff that entered your respiratory tract, your lungs, and you got to try to get it out. These cilia will kind of whip back and forth to try to drive that gunk, whatever was in there, upward out of your mouth. Okay, so that cilia will help. Also, you can think of like the fallopian tubes in women. So when the egg gets released, or the ovum gets released from the ovary, the egg can get guided towards the uterus with help of these cilia. Okay, so the cilia kind of help guide the egg closer towards the uterus. Okay, so that's the flagella and the cilia, which are just sometimes found in eukaryotic cells, not all though. Okay. All right, So moving on to the endomembrane system. So with the endomembrane system, we have basically it's just, endo means within the membrane, okay? So we're talking about a group of membranes and organelles that they're working together for a cause, okay? So it's basically, their job is to make, modify, and package up something for secretion out of the cell, okay? So if you think about like your entire body, your body's working, you know, you have different parts of your body working to, you know, supply other parts with things. So for example, in your pancreas is producing, the cells of your pancreatic cells are producing insulin. And what is insulin? Insulin is a hormone that is used to help regulate your blood sugar levels. And so the endomembrane system in your pancreatic cells are working together to modify, package, and transport insulin out of the cell. And so here are the components that are involved in that pancreatic cell to make the insulin. We have the nuclear envelope, we have the lysosomes, we have vesicles, we have endoplasmic reticulum, and we have Golgi apparatus. So we're going to go through each one of these structures within the cell. We're going to describe their functions and how they work in this endomembrane system to produce the end product. Okay, so and I'm just going to use insulin as an example. Okay, so let's start with the nucleus. Okay, so here is our big purple ball, so that is the nucleus. And you might think of the nucleus as like the boss of the cell. So the boss of the cell contains the cell's DNA. So that DNA houses the genetic information that has the instructions to make whatever the cell needs or whatever the body needs. So it's the entire blueprint of that organism. Okay, so if I can take the nucleus of any one of your cells and it contains the entire blueprint to make another one of you. Okay, so it has all the instructions to make another you. And so that DNA is housed on what we call chromosomes. So chromosomes, we as humans, we have 46 chromosomes. So those are just segments of DNA. And DNA is very long, okay? And we talked about that nucleic acid in last week with chapter 3. Or excuse me, that's from the Campbell book, chapter 2 from our book, the OpenStax book. But the DNA strand itself is really, really long. And to get it to be in a small nucleus, it needs to be kind of wound up. Kind of like if you have a big long piece of thread, it's better to kind of wind it around a spool, right? So you have a spool of thread that's nice and organized instead of a big wad of thread that just kind of knots and everything. And so what happens with that DNA, it gets wound around in a nice package, and it gets wound around protein in a nice package, which we call chromatin. So chromatin is just protein plus DNA. So chromatin is what makes up a chromosome. So chromosomes are made of chromatin, which is protein plus DNA. So you see here, these little spaghetti strings are just individual chromosomes. And if this was a human nucleus, there'd be 46 of these spaghetti strings, these chromosomes in there. All right? And they carry the instructions to make all the different proteins and things that the body needs. Okay, so there might be a segment of this chromosome here that codes for the insulin gene, for example, that codes for the insulin protein. So that's the nucleus. Now within the nucleus, you have the DNA that's just kind of sitting out here in the nucleus. You have this dark purple blotch, which is called the nucleolus. So the nucleolus contains something called rRNA. So rRNA, we talked about RNA earlier. So RNA is that ribosomal RNA, or excuse me, RNA is like DNA, is a nucleic acid that is very similar to DNA, except it's, it contains a different type of sugar, and it's single-stranded instead of double-stranded like DNA and so forth. There are different types of rRNA, one being rRNA. So rRNA stands for ribosomal RNA. And what does that sound like? Ribosomal RNA sounds like ribosome. And so rRNA makes up ribosomes. So it helps make ribosomes. So the nucleolus, its job is to make the building blocks of a ribosome. Okay, so it's making the rRNA, which makes ribosomes. Okay, so the nucleolus it's basically responsible for making the parts of a ribosome. And we'll talk about ribosomes in a little bit. All right. So we have the nucleolus that houses the building blocks of a ribosome. We have the regular nucleus part that houses the chromosomes that contains the DNA. And then we have a double membrane that holds the nucleus, which is the nuclear envelope. And that nuclear envelope, you can see there's little pores that, little pieces of RNA can squeeze through. We'll talk about that later on in chapters 9 and 10. But for now, we just have this nuclear envelope that's a double membrane that surrounds it. Okay, so that's the nucleus. All right. Now, when we're talking about the nuclear envelope, you can see how the nuclear envelope, it is, it's the boundary for the nucleus. But then attached to the nuclear envelope, it just kind of continues to this next membrane structure. So this next membrane structure is called the endoplasmic reticulum. So the endoplasmic reticulum is basically just this folds of membranes that continue off of the nuclear envelope. And this is where things are actually made. Okay, so the nucleus contains, it's like the boss of the cell. So it's telling, it's going to be telling the cell what to do. And right outside the nucleus is this endoplasmic reticulum, which I like to think of as like the factory floor. So this is where things are going to actually be manufactured. So it's these, folded membranes where things will actually get made. And so the first area of our factory floor is called the rough endoplasmic reticulum, otherwise known as the rough ER. Okay, and the reason why it's called the rough ER is because there's these little dots on it, which are ribosomes. And what are ribosomes? What do ribosomes do? If you remember, ribosomes are little protein factories. Okay, so protein factories, they're going to be making and manufacturing proteins. So if they're attached to the rough ER, they're going to be getting instructions from the nucleus, and they're going to be making and manufacturing proteins. Okay, so that's part of the job of the endoplasmic reticulum. So the rough ER part is going to be engaging with protein because of the ribosomes, be manufacturing proteins, and then that membrane part is going to be taking those proteins, modifying them, and also making phospholipids. Okay. And then you can see, continuing with the membrane part, we start losing the ribosomes, but we continue with the membrane. So this transitions into the smooth endoplasmic reticulum. So because it's smooth, without those ribosomes, we no longer have proteins being made, but we have other things being synthesized. So we have carbohydrates, we have lipids, and we have steroid hormones being synthesized by the smooth ER. Okay. So we have, a number of things being manufactured on this factory floor. So we have, in this section, we got the proteins and the phospholipids. Over here, we got like other lipids and carbohydrates and things being manufactured. Okay. So all that's being made through this complex series of membranes. In addition to making things, the Smoothie R has an added function, is that it can detoxify certain chemicals. Okay, so it can actually metabolize alcohol, it can store calcium, so it can do a couple other things in addition to manufacturing the carbs, lipids, and steroid hormones. Okay. So the boss says, hey, make some insulin. So this is where the Rough ER is going to get involved with starting to manufacture some insulin. Smoothie R, might get involved with helping adding some components to that that product. Okay, so we're manufacturing here, all right, with the endoplasmic reticulum. Okay. Next, what happens is we have this package from the manufacturing floor of the endoplasmic reticulum. This package will get pinched off by the membrane, and it will travel in a vesicle to what's called the Golgi apparatus. So the Golgi apparatus, think of that as like the shipping and packaging department of a factory. Okay. So this is where this little vesicle will merge with the cis face of the Golgi apparatus, and the contents of the package, the vesicle, will then get sorted, it'll get tagged, it'll get packaged up. So it goes through the Golgi apparatus where it gets modified, it gets added on, gets little receptors added to it. Tagged so that when it leaves the cell, the finished product can be delivered to where it needs to go. Okay, so if this was like an immature, like insulin hormone, it needs to get tagged, it needs to get, packaged up so that everybody knows that this is, insulin. So it's got to get packaged up into a box with a mailing label stuck on it. Okay, so that's what the Golgi apparatus is going to do, so that it can get shipped out of the cell through another transport vesicle and leave the cell as the finished product. So insulin can then be secreted from the cell directly into the bloodstream, where it can be picked up by, wherever it needs to go. Right? So you can see from start to finish how we end up with secreting something like the insulin hormone. Okay, so Golgi apparatus, think of like the shipping department, packaging department, the endoplasmic reticulums, the factory floor, the nucleus, is like the boss telling everybody what to do. Okay, and then we have these little transport vesicles that kind of move things around within the factory. Okay, and then we have other components, such as the lysosomes. So the lysosomes I like to think of as like the security guard / janitor of the cell. So their job is to keep things organized and clean inside the factory. Okay, so imagine you're on the factory floor and something breaks down, and so you got to get rid of it. So that's what the lysosome's going to do. So it's going to break it down and get rid of it for you. Or let's say that somebody enters the factory that's not supposed to be there. So security alert, little bacterium enters the cell. The lysosome is going to attack it and dissolve it. Okay, so lysosomes, they're like the security guard / janitor of the cell. So they're going to break down different things. So they'll break down nutrients like proteins, polysaccharides, nucleic acids. They can break down worn out organelles. So if something breaks down like a mitochondrium or a little piece of something, worn out organelle, they can break down to its most the macromolecules, so that it can get reused. They can attack invaders, that kind of thing. All right. And the way they do this, by the way, is inside the lysosome, there's hydrolytic enzymes. So hydrolytic, think of like hydrolytic, that's like HCl. HCl, hydrochloric acid, has a very low pH. So if you have a very low pH, you can basically dissolve things very easily. Okay, so those enzymes break down things. Okay, so that's an important job for the endomembrane system. And then finally we have the vesicles and the vacuoles. Okay, so these are the transportation part of the endomembrane system. So the vesicles and the vacuoles, they're basically the same thing, it's just there's difference in size. So vesicles are small, vacuoles are big. Okay, so vesicles are small membrane-bound sacs, so they're involved with storage and transport, whereas vacuoles are membrane-bound sacs that are larger than vesicles, that are also involved with storage and transport. The only real difference between a vesicle and a vacuole besides size is that vacuoles don't fuse with other membranes and other cellular components. So when you see, like in our endomembrane system, something going from the endoplasmic reticulum to a Golgi apparatus, carrying something and fusing with the Golgi apparatus, that would be a vesicle, not a vacuole. So vacuoles, they don't fuse with other membranes. Okay, so they're more just storing things and transporting things within a cell, but they're not fusing with anything. All right. So again, vesicles very important with our endomembrane system to help move the products of our endoplasmic reticulum to our Golgi apparatus, and then from the Golgi apparatus to the plasma membrane. All right. So let's take a look at our visual connection here. Now, if you take a look at this Golgi apparatus, there's a cis face and a trans face. So why does the cis face of the Golgi not face the plasma membrane? So the cis face, by the way, is the receiving end of the Golgi apparatus, whereas the trans face is the end that transmits. Well, the cis face is receiving basically from what? From the rough ER. And the rough ER is attached to the nucleus, which is towards the center of the cell. Okay, so it doesn't make sense for this to be switched. So the Golgi apparatus is not receiving anything from the plasma membrane. It's receiving from the interior of the cell. It's receiving from the smooth ER or the rough ER. It's not receiving from the plasma membrane. So the Golgi apparatus is the packing and shipping department. So it's going to be shipping things towards the plasma membrane from the trans face. Okay, so it doesn't make sense for the receiving end to be facing the plasma membrane because it's the shipping department. All right. So the cis face faces the endoplasmic reticulum, the interior of the cell. All right, so we talked about our endomembrane system. So now we know how we make insulin secreting from the cell. So we have the boss of the cell, the nucleus. We have the rough ER being rough because it makes and manufactures the proteins and the phospholipids. Smooth ER manufactures the carbs and the lipids and steroids and other things, detoxifies things, stores calcium. We have the transport vesicles that transport things in between structures. The Golgi apparatus is the shipping center, so it helps modifies, packages the products, and then ships them out of the cell. And then we don't have our lysosomes. Those are the ones that are keeping things clear and clean and secure. Right. So moving on to the ribosomes. And by the way, you'll notice that I like to create these little analogies for things just because it helps us remember things. So I think of the nucleus as the boss. I think of the endoplasmic reticulum as the factory floor. I think of the lysosomes as the security/janitors of the cell. So you might want to do the same thing and create like your own little analogy. Okay, so it doesn't have to be like a factory. It could be like a city. So the city, you know, the mayor would be the nucleus. Okay, the streets could be the cytoskeleton or something like that. Okay, so we have our ribosomes. So we already talked about the ribosomes being the protein factories. So they're the ones that carry out the protein synthesis. And remember, what are ribosomes made of? So we talked about that being made of rRNA. So rRNA is made in the nucleolus of the nucleus. And this is what a ribosome looks like. And ignore all these weird, I just got this random picture from somewhere. So it's kind of hard to read. But you have this large subunit of rRNA and this small subunit of rRNA. So that's what a ribosome looks like, okay? And we'll talk more about this whole process later on, but for now you can kind of see here's like a little amino acid chain forming off of this ribosome. Okay, so ribosomes, they're protein factories. So here you have an amino acid chain being formed off of this ribosome. Now we have two types of ribosomes. One we just talked about with the ribosomes attached to the rough ER. So those are the bound ribosomes. So they're making the amino acid chains, the proteins. And so those proteins, they get used for secretion out of the cell. Okay, whereas a free ribosome, those are floating freely within the cytoplasm. So those proteins will be used within the cell, okay, because the cell needs its own proteins for its own usage. So bound ribosomes attached to the rough ER, which will then get secreted from the cell. Free ribosomes make proteins for use within the cell. Okay, so that's the difference between the two types of ribosomes. Bound ribosomes make proteins to be secreted. Free ribosomes make proteins to be used within the cell. All right, so now we're just going to look at some of the organelles that are found within the cell that are not attached to like the endomembrane system. So the first one is the mitochondrium. And the mitochondria, that's that little kidney bean looking thing. So this is what it looks like under a microscope. And it is, think of that as the powerhouse of the cell, or the little, basically the power plant to the cell. So this is going to be where the energy is coming from. So this is what generates the ATP that powers the cell. And we'll talk about in chapter 7, cellular respiration. So cellular respiration is that process where we take nutrients, such as glucose, and we extract cellular energy from that. And so that's the site of that process, so the mitochondria. So the mitochondria is carrying out cellular respiration, resulting in the production of ATP. So ATP is the cell's energy carrying molecule. Okay. And what's interesting about mitochondria is that we actually think mitochondria at one time was a prokaryotic cell. So we think that an ancient eukaryotic cell actually engulfed one of these prokaryotic cells, and instead of just digesting it, it became incorporated in the eukaryotic cell. And the reason why we believe this was an ancient prokaryote is because the mitochondria has its own DNA. as well as its own ribosomes. So it actually has all the components of a cell. It has a plasma membrane, it has cytoplasm, it has its own little circular piece of DNA, which is similar to bacterial DNA. It actually divides on its own, and it has ribosomes. So it supports, like if you read in the textbook, it supports this idea of what's called the endosymbiotic theory, where we think this was an ancient prokaryote at one time. Okay. And this can also be used in like ancestral, if we want to look at like our looking at human history, we can trace, because you inherit your mother's mitochondrial DNA, because when sperm meets egg has mitochondrial DNA in it. Sperm doesn't have any mitochondrial DNA, and so that doesn't get transferred. It's just the nucleus of the sperm. And so you actually have a little bit more of your mother's DNA because you inherited your mother's mitochondrial DNA. So we actually can use mitochondrial DNA in human ancestry, so we can actually trace some of that. And, you know, take a look at where like humans have traveled and so forth in our human ancestry. But anyway, so you're a little bit more of your mom than your dad because of your mom's mitochondrial DNA. But anyway, so that's an interesting thing. Off topic. So mitochondria, that's the powerhouse of the cell. So it's helping provide the ATP. And we'll go into that in next week, I believe. I can't remember. Moving on, we have peroxisomes. So peroxisomes, if you look right here, here's the kidney bean, by the way. So that's your mighty mitochondria. Has its own little DNA. Okay. Here is your peroxisomes. So the peroxisomes, their job is to help detoxify things. So they will carry out some oxidation reactions. And we'll talk about oxidation and reduction reactions later on, but they help break down fatty acids and amino acids. So remember how when you're taking in nutrients, sometimes you're taking in things that are big, okay? You're taking in like complex carbohydrates, those polysaccharides, or you're taking in, triglycerides that have, a bunch of fatty, tri means 3, so 3 fatty acids, but you only need 1. So you got to break them down. So these peroxisomes can help break things, big things down into smaller components. So they can break down things into smaller components like the fatty acids and the amino acids, so that you can just use individual amino acids to build other proteins from them. They can also detoxify things like alcohol, and because they contain some enzymes, their byproduct tends to be hydrogen peroxide. So hydrogen peroxide is H2O2, so that can be further broken down into water. All right, so it's a useful organelle to help detoxify things into something that is not toxic in the least. Okay, so they break down big things into small things and detoxify things. All right. Okay, so now we're going to get into animal cells versus plant cells. So the difference between an animal cell and a plant cell. We talked a little bit about this already at the beginning. So we'll go through some of these. So we know that plant cells, they have the cell wall that surrounds. We'll talk about that with the plasmodesmata. We'll talk about the chloroplast, the site of photosynthesis. We already talked about the central vacuole, so that's that big, large place. And then I don't remember if we talked about plasmids, but they carry pigment. So think of like a flower with its bright colors. So that's due to the plasmids. Animal cells, we talked about the centrosomes, so that's part of the cytoskeleton. We talked about lysosomes already. We didn't talk about centrioles, but that's part of cellular division. So we'll talk about more of that in chapter 9 and 10. So we'll look at some of the differences between plant and animal cells, starting with the plant cell's cell wall. So cell walls, so they surround the cell membrane or plasma membrane. So you can see here, I think in yellow is the cell membrane. So this is the big green part. So that is the cell wall. We don't have the cell wall. And it's a good thing because it's very rigid. It would make it impossible for us to move. And for us, this is something that we can't digest, right? Because it's made of cellulose. So cellulose is the that polysaccharide that makes up the cell wall in plants. It's chitin in fungi. And it has 3 jobs, that cell wall. It helps protect the cell because plants, they, can't protect themselves by running away. So it helps protect the cell from, bacteria and stuff like that. It provides structural support. Okay, they don't have, it helps hold it upright. And then it gives shape to the cell. So this week in the lab portion, you'll see the plant cells under a microscope, and you'll see they're all little rectangles. Whereas animal cells, they're all weird shapes. Okay, so that gives the cell that rectangular shape. So cell walls, they help maintain structure, shape, and protection for the cell. Chloroplasts. So chloroplasts are something that we don't have because we don't photosynthesize. So chloroplasts, they are the site of photosynthesis. So this is where the plant has these things called thylakoids, and inside the thylakoids is a pigment called chlorophyll. So that green pigment chlorophyll helps absorb sunlight, and that's light energy from the sun helps the plant convert carbon dioxide from the air and water from the soil into glucose. Okay, so it can take CO2 and H2O and convert it into C6H12O6 glucose. So it can make its own glucose through having one of these structures, the chloroplasts. So we can't make our own glucose, right? We have to eat it. So chloroplasts, plants have that in order to make their own glucose. And something interesting about the chloroplasts, just like the mitochondria, we think that the chloroplast was an ancient photosynthesizing bacteria at one point in time, or prokaryote at one point in time. Because like the mitochondria, it has its own DNA and its own ribosomes. So we think that, an ancient cell engulfed photosynthesizing prokaryote, and for some reason it didn't digest it, but it became incorporated, and that cell started photosynthesizing and using that that structure, the chloroplast, to make glucose. All right. So again, chloroplasts used to make glucose through photosynthesis, and then of course that glucose can then be used in the mitochondria of the plants for cellular respiration. So a lot of students forget that plants, they also have mitochondria. They have both. We don't have chloroplasts, but plants do. Plants have both mitochondria and chloroplasts. And then the central vacuole. So we have little vesicles and we have small vacuoles, but we don't have this large vacuole. So that large central vacuole is a storage compartment for plants. So think about plants. They can't get up and walk around like we do. So if they if we're thirsty, we can go get some water, but plants, they can't do that. So they rely on having water reservoirs inside them. So during the summertime, they need to have their storage vacuoles filled with water to get them through our long, relatively dry summers. They can also use their vacuoles as a storage compartment. So again, think about the potato. So the potato tuber uses its vacuoles to store that starch. Okay. So that micromolecule degradation, so it can use it to store things. Okay, so just a way that plants can store things away because it can't exactly go get it itself. And then we have extracellular matrix. So this is something that was not mentioned in our list. So this is something that we have for animal cells. So here is our plasma membrane. Here's the inside of the cell, and then here's the outside of the cell. And you notice on the outside we have all this weird stuff hanging out. So this is actually not considered part of the cell because it's outside of the cell. Okay, so this is extracellular, outside of the cell. And it's attached to the cell, so that's why it's called, kind of considered, part of this lecture. So it can be secreted from the cell. We can have collagen fibers. So collagen is, kind of protein and fat. Glycoproteins. Glyco is sugar, protein. Proteoglycans. Okay, so proteoglycan contains basically a polysaccharide, proteins, and carbohydrates. So all of this stuff gets secreted from the cell but stays attached to the cell. And what it does is it holds cells together to form tissue. So remember in our life's hierarchy, we have cells forming tissue, tissue forming organs, and so forth. So cells, they can join together with this extracellular matrix. Okay. It also allows cells to communicate with each other. So cells can, if they have this extracellular matrix, they can easily, if they're kind of stuck close together, they can communicate with each other and anchor to each other, which can also provide mechanical protection. All right, and then the final area of our video that we're going to talk about are intercellular junctions. So just like the extracellular matrix, this occurs outside of the cell, but it's important because it allows for communication and allows cells to stick together and things like that. So we're going to start out with, first of all, I guess here's the definition, by the way. So intercellular junctions, so it's a way that they can communicate with each other by direct contact. So we're going to start out by looking at plant cells and their intercellular junction, which is a plasmodesmata. And then we'll move to animal cells, and there's 3 of them. We'll learn about tight junctions and gap junctions and desmosomes, otherwise known as anchoring junctions. So starting us out are plasmodesmata. So plasmodesmata or plasmodesma occur, if you recall, with our plant cells. Our plant cells have that extra cell wall. And so because of that extra cell wall, which allows for structural support and protection, plant cells, they need a way that they can exchange, different nutrients and communicate with each other and things like that. And so they have these little gaps in between the cells. And so those gaps are what we call the plasmodesmata, or plasmodesma is the singular. Okay? So plasmodesmata is just that gap that allows for transport of nutrients and things like that to flow between two plant cells. All right. So that's intracellular junction for plant cells. Moving on, for intracellular junctions for animal cells, we're going to start with our tight junctions. So tight junctions, it's almost like Velcro. So imagine this is Velcro. So these tight junctions, they form a watertight seal. And the way that they do that is they have proteins holding the two cells together. And so this creates this watertight seal that prevents, again, water from passing through. And it holds the two cells together. Okay, so a tight junction holds the two together. All right. So, and again, it's like Velcro, so they can come apart, but their job is to create that watertight seal. And then we have the desmosomes. So desmosomes are also known as anchoring junctions. And an anchoring junction, think of like a weldering, or I don't know, I'm not into like home fixing projects, but like a rivet. They are very strong. So a desmosome, it's going to anchor, or really hold the two cells together very strongly. Okay, so it's an anchoring junction. It holds them together very strongly. So in a sheet-like faction. So in this case, like if you tore like an anchoring junction or desmosome, that would hurt because these are really strong connections. So tight junctions, they're kind of create a watertight seal. Anchoring or desmosome junctions, they're going to be very strong junctions that hold the cells together. And you can see the intermediate filaments sticking together. And then finally we have a gap junction. So gap junctions, imagine those are kind of like the plasmodesmata of plant cells. But in this case, we have them for animal cells as a gap junction. So there are these little gaps that allow for things to be passing through easily from one cell to another. In this case, you can see what it looks like when it's closed and what it looks like when it's open. So two cells can pass ions, nutrients, and other substances, so they can communicate back and forth. All right. So gap junctions allow for things to pass through. Desmosomes, they're anchoring two together like rivets, and then the tight junctions are watertight seals. All right, so let's do some review questions. So which of the following is not a component of the endomembrane system? Is it the mitochondrium, the Golgi apparatus, the endoplasmic reticulum, or the lysosome? So which one of these is not part of the endomembrane system? If you said the mighty mitochondrium, I would agree with you. So remember, the mighty mitochondrium is a separate organelle that is the powerhouse of the cell, but it's not necessarily part of the endomembrane system. So the endomembrane system includes the nuclear envelope, the rough ER, the smooth ER, the vesicles, the lysosome, and then you can even see, say, maybe the plasma membrane, because the vesicles fused with that. And the Golgi apparatus, I forgot about that because it's not shown here. All right, so the mitochondrium is not part of the endomembrane system. Right? So by the end of this, so you should have, your chart with the different organelle or cell component, and then the function, and then perhaps a picture. In your textbook, this is, I believe it's table 3.1, has a good summary of all of the different cellular components, as well as, gives you the function, and it tells you if it's present in the prokaryotes, in animal cells, and if it's present in the plant cells. So this is a good thing to kind of use as a review as well. This would be good to maybe create flashcards out of, or you can kind of make a blank version of this chart with just the cellular components, and then you can kind of fill it in from memory as a good review as well. So you can see we've gone over, I think, all of them. We didn't specifically say like centrosomes, we just said it's part of cell division and it's made of microtubules, but we didn't really say that. Well, we did say it's part of animal cells. And then it continues. I had to cut it. So it continues. It's a long chart. We talked about lysosomes. We talked about flagella, cilia. So we talked about all of them. Yep. So this is a good summary chart that says everything. So if you didn't get everything down in your notes, use this to kind of fill it in for you. All right. So we are done with 3.3 eukaryotic cells. So by now you should be able to describe the structure of the plant and animal cells. So again, that's why I had you kind of draw the different organelles. You should be able to state the role of the plasma membrane. You should be able to summarize the function of the major cell organelles, as well as describe the cytoplasm and extracellular matrix. Okay. So our final video for this unit, we're going to specifically focus in on the cell membrane. So if you didn't get that role of the plasma membrane, don't worry, because we're going to go into detail in 3.4.